For my thesis, I investigated the potential role of MAD2 in the spindle assembly process. The cell cycle is tightly regulated to ensure that the genetic material is properly duplicated and segregated into daughter cells. There are three main checkpoints, and each one has an important role, but I'm going to focus on the spindle checkpoint. This checkpoint prevents the onset of anaphase until all chromosomes are properly attached to the spindle. It is known that MAD2 plays a role in regulating the spindle checkpoint. When it is present, the spindle checkpoint is functional, as seen in the image to the left, and the chromosomes segregate properly. However, if MAD2 is missing or mutated, the spindle checkpoint becomes non-functional, and aneuploidy can result, as indicated in the rightmost image. We know MAD2 is important at the spindle checkpoint, but the NanoSlab suspects that it could be involved even earlier in the spindle assembly process, because they recently found that 80% of the observed meiotic cells initially assembled their spindles incorrectly. As a relatively simple tool that can edit genes, CRISPR is what makes my project possible. It stands for Clustered Regularly Interspersed Short Palindromic Repeats and has two main components: a guide RNA that recognizes the targeted gene, and the Cas9 protein that actually creates the double-stranded break. As the cell tries to repair the break, it can do so in a way that causes the gene to be knocked out. For my project, the guide RNA was designed to target a sequence within the MAD2 gene, where Cas9 would cut and hopefully generate a MAD2 knockout. This is the part of the MAD2 gene that we targeted for amplification through the use of primers. It is located towards the beginning of the entire gene and is early in the gene coding sequence. The important parts of the segment have been highlighted. The PAM sequence that the Cas9 needs to recognize before binding or cleaving the DNA is highlighted in green. The sequence the guide RNA will bind to in pink, the EcoR5 site in purple, and the primers in light blue. The CRISPR cut site is indicated by the three base pairs shown in white. In addition to being within a translated region, the guide RNA was designed to include an EcoR5 restriction site that was three base pairs upstream of the PAM sequence. Because CRISPR cuts 3 to 6 base pairs upstream of the PAM, any mutations to the MAD2 gene would occur within the EcoR5 restriction site. This would effectively disrupt the gene and cause the EcoR5 site to become unrecognizable. Thus, by later digesting MAD2 with EcoR5, we can observe if a CRISPR MAD2 knockout occurred. When screening for a MAD2 knockout, there are two possibilities that can be observed. If CRISPR modification did not occur, EcoR5 digests the gene into two segments because it can recognize the EcoR5 cut site. This can be seen in the graphic to the left. However, if CRISPR modification did occur, the MAD2 segment will remain in one piece, because as the cell attempts to fix the double-stranded break created by Cas9, insertions or deletions cause the EcoR5 cut site to become unrecognizable. When the EcoR5 restriction site is unrecognizable, EcoR5 cannot make the cut. This is depicted in the graphic to the right. The presence of one or two DNA segments is best visualized on a gel. Before the samples can be run on a gel, however, several steps have to be taken. First, DNA is extracted from the plants. Then, MAD2 is amplified via PCR prior to being digested with EcoR5. The digested samples are finally loaded onto a gel, where they separate based on DNA fragment size. Two bands around 255 and 245 base pairs are expected if there is no CRISPR modification, whereas one band around 500 base pairs is expected if there was CRISPR modification. These are two gels that I ran. Each gel has a ladder and a positive control of wild-type MAD2 that should get cut since it lacks the CRISPR components. The gels contain plants from two different plant lines that result from different guide RNA transgenic plant lines being crossed with different Cas9 transgenic plant lines. The NanoSlab has 20 transgenic guide RNA plant lines and 3 Cas9 transgenic plant lines. Both of these gels did not indicate a MAD2 knockout and are representative of the other 179 plants I screened. In the future, screening for a MAD2 knockout should continue alongside the measurement of guide RNA and Cas9 levels, because it is possible that the expression levels of guide RNA and Cas9 differ between plants. The expression levels can differ between transgenic plant lines, even though they are genetically identical, because the transgenes could have different epigenetic marks that impact expression. So, by measuring expression level, we can select plants with higher expression levels of the guide RNA and Cas9 components, since they could be more likely to generate a MAD2 knockout. By running these two approaches simultaneously, we can hopefully speed up the discovery of a MAD2 knockout so that live imaging can be used to investigate if MAD2 is involved in spindle assembly. And eventually, these results could aid in future research on preventing aneuploidy, which is behind cancer, infertility, miscarriage, and birth defects. I would like to extend many thanks to Professor Natalie Nannis, the Hamilton College Biology Department, Alex Chalk, Cassie Pontius, Emnit Sase, and Grace Godwin, because without them, this project would not have been possible.